Assalamu alaikum students. Before going to our educational topic, please subscribe my channel Muntzir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic, that is qualities of enzymes, properties of enzymes. Dear students, this enzymes topic is also known by the name of characteristics of enzymes, which is the common name of this topic. This topic is explained by the means of different points, which we will study now one by one. First point is that enzymes are globular proteins and are protein in nature, which means that the enzymes are made up of globular proteins that are rounded circles of amino acids. A large number of amino acids are present in the enzyme's body, and these amino acids are arranged in this way that these aggregate to form tertiary protein configuration. So enzymes consists of tertiary and globular structure of proteins. Globular means round circles like, that is the shape of amino acids, and tertiary means when a polypeptide chain folds upon itself like this, then it will be tertiary structure of protein, and this is the same structure that is present in the enzymes. Tertiary as well as quaternary structure of proteins are associated with not only to proteins but also to enzymes. So we can say that an enzyme is made up of proteins possessing tertiary as well as quaternary structure. Students, I will prepare a video of protein structure in detail later during next upcoming videos, because it is a separate topic that what is a tertiary structure and what is a quaternary structure of protein. As it is said that when more than 50 amino acids will join together, then these amino acids will prepare a polypeptide chain that will be processed into a protein. But sometimes it is said that not only more than 50 amino acids are required, but quaternary structure is also required for the preparation of a protein. So when more than 50 amino acids will present as well as quaternary structure will present, then it will be a protein. So collectively we can say that enzymes are protein in nature and are made up of globular proteins as well as tertiary and quaternary structure of proteins. Second point is about the enzyme's job, that what is the job of an enzyme. Dear students, enzymes speed up a chemical reaction, catalyze a chemical reaction. It has also discussed already in the previous videos. As when reactants like reactant number 1 and reactant number 2 combine to prepare a product, so if this catalytic reaction happens in 5 hours without enzyme, then in the presence of an enzyme it will take just 5 seconds only. So we can see that in the presence of an enzyme a chemical reaction takes place rapidly in a short time period. So enzymes accelerate a chemical reaction in an excellent way. Third point about enzymes chemical reaction is that enzymes have no effect on the chemical and functional nature of an end product, because enzymes are not being used up during a chemical reaction. For example, when these two reactants, reactant number 1 and reactant number 2, react with each other in the presence of an enzyme, then these will synthesize a product that is being prepared here. So when after the product formation, we will see that the enzymes will remain the same as it was before. There is no chemical change, there is no physical change in the product and in the enzyme. So we can conclude that the enzymes just speed up and accelerate a chemical reaction without being used up during their catalytic activity. So now when they are not utilized during the chemical reaction, then they have not any effect on the chemical nature and physical properties of the end product. Fourth point is that how much amount of an enzyme is required to catalyze a particular chemical reaction. The answer is that we will not require a large amount of an enzyme to catalyze a chemical reaction to change a large number of substrates into their respective products. But only a small amount of an enzyme can bring about large changes in the large amount of the substrates. For example, a single enzyme molecule may split 100,000 substrate molecules in just 1 second. So we can conclude that only a tiny amount of an enzyme is required to bring about large changes in the specific amount of a substrate required amount of a substrate. Fifth point is about the specificity of an enzyme, which tells us that enzymes are specific, very specific. Specific means an enzyme can act upon only a single type of substrate or a group of related substrates in related chemical reactions. In this way, we can say that a single enzyme can't work upon the different natured substrates, but it can work only upon its related substrates. For example, cellulase enzyme can digest only cellulose material, which is a carbohydrate. So cellulase enzyme can't digest the proteins, fats or nucleic acid like food materials. So specificity of an enzyme refers to that lipids digesting enzyme, namely lipase, functions upon only phospholipids, whereas sucrase enzyme work upon sucrose sugar only, and maltase enzyme can act upon maltose sugar only. So maltase and sucrase can't function upon phospholipids, and lipase enzyme can't function upon sucrose and maltose sugar. So we can say that every enzyme is specific for its specific substrate but not for other food groups. But if you want to understand this specificity of an enzyme in more depth, then see, this is the lipase enzyme that we have. This is the protein food group, this is the lipids food group, and this is the carbohydrate food group. So out of these three food groups, lipase enzyme will act only upon lipids food group, because this lipase enzyme will be specific only for its specific substrate, that is lipids food molecule. So it will act upon lipids food molecule and will digest it. At the sixth point, we will study about the sensitivity of an enzyme, which means that the enzymes are very specific to a bit change into a small change in pH, temperature, and substrate concentration. Namely, an enzyme requires a specific pH value, specific temperature range, and specific number of substrate molecules upon which that enzyme will act. This specific pH value is known as optimum pH. Required temperature is known as optimum temperature, and so on. For example, the temperature of human body is about 37 degrees centigrade or 98.6 Fahrenheit. So pepsin enzyme will function at about 36 to 37 degrees centigrade temperature in the humans very excellently, but above or below this temperature range, pepsin enzyme cannot perform its function. For example, during the fever, when we are suffered from high fever, we can't eat the bread or any other heavy diet because our body temperature become above or below than 100 degrees centigrade. At this very high temperature, students, pepsin enzyme can't work its job properly. That is the way that it is said that enzymes are sensitive, because if they were not sensitive, then they can work at any temperature in the living organisms. That's why it is said that enzymes are sensitive to heat, because the enzyme's activity increases with the increase of temperature up to 50 degrees centigrade. So when a body temperature becomes up to 50 degrees centigrade, then there happens initial rise in the enzyme's activity above this temperature, that is up to 50 degrees centigrade. But this increased enzymes activity is soon followed by a decrease in activity, decline in activity, because above to certain range of body temperature enzymes activity increases, but after certain range of increased activity the enzymes activity begins to decrease, because the enzymes are denatured at very high temperatures such as 70 to 100 degrees centigrade. But some such enzymes also present which can withstand temperature of about 100 to 120 degrees centigrade for long periods in dry conditions. Such conditions are present in some spores and dry seeds. Students, this is the enzyme sensitive behavior at high temperature. Like high temperatures, enzymes are also sensitive to low temperatures because they are inactivated at very low temperature, but they are not destroyed at 0 degree or below than 0 degree centigrade. In addition to these high or low temperature range, low temperature problems, enzymes also require a specific pH value. Consider that different enzymes are working in different parts of our bodies. If we take the same pepsin enzyme which performs its job in human stomach, it works at a pH of about 2, 2.1, 2.22 around. As it is an acidic enzyme, so it functions at acidic pH betterly. That is about 2 pH value. Above or below this pH, it can't perform its function. Why? Because enzyme will be pH specific. Students, if we consider the intestinal enzyme trypsin, which works on basic medium alkaline medium, it can perform its function above than pH of 6 pH value. Similarly, catalases show optimum catalytic activity in a neutral pH value. Amylose shows best activity in a neutral or slightly acidic medium, whereas sucrase and lipase show best activity at acidic pH value. So students, every enzyme possesses an optimum pH value whose activity decreases or increases with an increase or decrease in the pH value. So it is a major issue with the enzymes that they are specific. They require optimum temperature and optimum pH value. Dear students, in addition to temperature and pH values, enzymes are also influenced by radiations. If radiations are allowed to hit a living organism, then its inner enzymes will also be affected, denatured, because due to this exposure of radiations enzymes are destroyed, denatured by the radiations of lower wavelengths like ultraviolet rays and x-rays. Next, seventh point is about the enzymes and cofactor. Dear students, as we have known that enzymes are made up of proteins themselves, so they are efficient and important to perform their catalytic activity. Because an enzyme can't start the reaction, it can only accelerate the chemical reaction. So some enzymes, not all the enzymes, but some enzymes must require a non-protein part whose name is cofactor. In my previous video, which is the second part of enzymes lecture, I have described with much more detail cofactor. That a cofactor comes and is attached to the enzyme to prepare a complete enzyme which perfectly performs its function. If you have not watched that video till now, then you can watch it now. From it you can clearly understand that the cofactor, which is the helper of an enzyme, attached to it and helps it in its catalytic activity. Eighth point is about the activation energy. Dear students, activation energy is that minimum amount of energy which is required to start a chemical reaction. Students, keep this important point in your mind that activation energy is required to start a chemical reaction, to initiate a biochemical method, not the enzymes. Enzymes just catalyze the chemical reaction, not start. and activation energy is needed to start a chemical reaction, to initiate a chemical reaction. And enzymes lower the activation energy during the reactions. A reaction can take place in the absence of an enzyme, but it needs higher amount of activation energy. We can also understand this mechanism with the help of this helping diagram. In this graphical diagram, time is present at the x-axis, whereas amount of energy is present at the y-axis. Two reactants A and B are present at this initial state. Students, in the absence of an enzyme, these reactants require a higher amount of activation energy to change into a product. This curve shows that higher amount of activation energy is required to change the reactants into product, whereas in the presence of an enzyme, these reactants will require a lower amount of activation energy to be changed into a product. This activation energy is without an enzyme, whereas this activation energy is with an enzyme, and this gap will represent the overall energy change between the reactants and the product. So we can prove from this diagram that enzymes lower the activation energy to initiate to start a chemical reaction. This reaction will take place rapidly in the presence of an enzyme but at the same temperature. Ninth point is about the aqueous environment. We can understand it with the help of this diagram. It is our cell which must possesses protoplasm. About 90% of a cell consists of water content cytoplasm. So this water environment inside the cell provide aqueous medium to the enzyme for its better and proper job, because when aqueous medium will available to the enzyme, then enzyme will dissolve in this cytoplasm's aqueous medium, which is very necessary to the enzyme's job. At the 10th point, we will discuss about the reversibility of enzyme action. Students, most of the reactions which are catalyzed by the enzymes are reversible. Therefore, an enzyme can catalyze a chemical reaction in both directions. So the reaction gets an equilibrium state in a short time. For example, in the guard cells of the stomata, an enzyme starch phosphorylase converts the starch and inorganic phosphate into glucose monophosphate. This reaction is reversible. Therefore, the same enzyme starch phosphorylase converts glucose monophosphate into starch and inorganic phosphate again. So like this, majority of reactions are reversible. Students, the direction of reaction depends upon several factors like pH and chemical potential of both the reactants. When the chemical potential of the product is less than that of reactants, then the reaction takes place only in one direction. It will not be reversible. Students, it is a fact that synthesis of starch, proteins and fats are unidirectional enzymatic reactions which are irreversible. Such reactions have two different sets of enzymes. One enzyme is utilized for the synthesis of these compounds and other enzyme is utilized for their breakdown, for their splitting. At the 11th point, we discuss the damaging effect of enzymes. Students, if enzymes are allowed to start its catalytic activity, to start its function at the malfunction site, wrong site, then they will become harmful to that mismatched site. For its better understanding, we see the pepsin example. Students, as pepsin is present in stomach and it is a protein digesting enzyme. So when it is secreted from the stomach wall, when it is secreted, then it can harm, destroy not only the stomach wall but it can also destroy the cell's internal structure, cell's internal surface of the stomach. Students, because of this damaging effect of the pepsin, it is not directly secreted from the stomach wall, but it is secreted in an inactive form, that is pepsinogen, by the stomach cells. This inactive form pepsinogen is enclosed in membranous bounded organelles, membranous bounded bodies that are called lysosomes. Due to this inactive state, stomach safe from the damaging effect of pepsin enzyme. At the 12th point, we study about the attachment of enzymes. Students, some enzymes float in the cytoplasm, but mostly enzymes do not float in the cytoplasm because they are attached to the membranes in the cell. For example, the enzymes of mitochondria and chloroplasts, they are mostly attached to the membranes but not float in the cytoplasm. Next is the 13th point, that is about the product inhibition. Students, during product inhibition, we suppose that when a large number of products are stored or become abundant in a cell after enzymes catalytic activity, then the storage of product can inhibit the enzyme's function, its job. Students, this blockage of product formation is very crucial for cells because it is a self-regulating mechanism of reactions progression and maintains the product concentration in the cells of a body at a suitable level, at an acceptable level. For example, it is an obvious point that when required amounts of products are achieved, then enzyme will stop to perform its function. Thus, enzyme's activity is inhibited with the reservoir of products in a cell. Next is the 14th point, that is about allosterism. Dear students, certain enzymes have some specific sites which are known as allosteric sites. Some specific bodies come that are called effectors and bind to this allosteric site. This binding of effectors create a change, bring a bit change in the enzyme structure in its body shape. This whole experience during which an effector binds with an enzyme at its allosteric site is called as allosterism. Students, this unique phenomenon of allosterism is shown by many different enzymes. Next, during the 15th and last point, we try to understand the in vivo and in vitro terms about the enzyme's function. Students, in vivo is the study of enzyme. For its better understanding, suppose it is a cell of an organism. An enzyme is present in the cell. If we study this enzyme inside the cell, then it will known as in vivo study. On the other hand, if we study an enzyme outside the body cell in the laboratory glass or test tube, then it will come during the in vitro study. That's why for in vivo study, we will say that inside the cell or inside the living organism's body, or for in vitro study, an enzyme is discussed or studied outside the cell in some laboratory equipment. So students, it is our topic today, characteristics of enzymes. That is very crucial topic, often comes during annual examination. So prepare it very well by understanding the basics and concepts of all the points. At the end, I want to encourage you that be happy yourself and keep happy others. Students, for saying it is a very short thing, but it has very excellent consequences. Lastly, thank you for watching this your video.